Alright. Okay.
Dear students, in this video we are going to see yet another topic, microbial growth. The topics to be covered in this video under this topic are: number 1, difference between human growth and bacterial growth; number 2, modes of bacterial cell division; number 3, bacterial growth curve. Let us see the first one, the difference between human growth and bacterial growth. In humans, the term growth refers to an increase in size. For example, growing from a teeny newborn baby to large adult. Although bacteria do increase in size before cell division, but we cannot identify it as they are microscopic. Microscopic in the sense we cannot able to observe them with our naked eyes. We need microscope in order to observe them, as they are microscopic. So bacterial growth refers to an increase in the number of organisms or cell number rather than increase in cell size. So bacteria grow and divide. Bacteria grow and divide by binary fission. This is important. This is a binary fission, a rapid and relatively simple process. Let us watch this bacteria how rapidly divides. We can observe with the help of a video that was recorded with the help of a electron microscope. Let us watch. Bacteria reproduce very simply and rapidly by doubling its contents and splitting in two. Just one bacteria that divide every 20 minutes could produce 5,000 billions and billions in one day. Now let us see how bacteria reproduce itself. Modes of bacterial cell division. Bacteria can divide by number 1, binary division. Second one is going to be budding. Third one is going to be fragmentation. Let us see the first one, binary division. What is this binary division or binary fission? Binary fission is the most common mode of cell division, in bacteria. In this mode of cell division, a single bacterial cell grows transversely with the synthesis of chromosomal DNA. That means a bacteria doubles its DNA content. And then what happened once it transversely increases its chromosomal DNA or bacterial DNA, then what happened? A transverse septum appears in the middle of the cell body that divides the bacterial cell into the two with a distribution of chromosomal DNA, ribosome and other cellular machinery. Let us see how it happens with the help of a diagram. This is what is called binary division. We have seen this one, binary division. In binary division, what happened? This is bacterial cell. Inside the bacterial cell, you can see the DNA present, and around the bacteria there was a cell wall and a plasma membrane. And let us see what will happen here. So duplication of chromosomes is occurring. Duplication of chromosomes, that means doubling of DNA is happening. This is also called as replication process. The replication, this is one DNA and this is the second DNA that is formed inside the bacteria. And then what happened? A transverse septum, this is what the transverse septum is formed, and the DNA is distributed here to here. DNA is distributed and then a transverse septum is formed, and through this septum what happened, this bacteria divides into two. This is called, now these two become individual bacterial cells. This is called binary division or binary fission. This is the most common bacterial division we have seen regarding that. Binary division is the most common mode of cell division in bacteria. See here, another thing is the cell grows transversely. That cell grows with the synthesis of chromosomal DNA. A transverse septum appears in the middle of the cell body that divides a bacterial cell into the two, with the distribution of this DNA, ribosome and other cellular machinery. That is what we have seen in this diagram. This is what here. This is the bacteria, and replication takes place. Doubling of chromosome, doubling of bacterial DNA is happening here, and whatever the doubled DNA is segregated into two different cells, and now a septum is formed, and two different cells are formed. Two individual cells are formed by this one. And another we have seen, what is that one? Binary fission. The another one is called budding. In this mode of cell division, bacterial DNA divides to form two copies, as usual in the same in the earlier one also. Replication has taken place. Now what happened? The sister chromosomal, the DNA moves into one side of the cell. It is not distributed. The sister chromosome moves into one side of the cell. Then this portion of cell protrude from the main body to form a bud. So that means a bacterial cell give out a bud, and into the bud what is transferred? This DNA is transferred. Eventually bud grows in size and gets separated from main cell to develop a new cell. This process is called, what is that one? Budding. This is called budding process. And this budding process, you could be able to see, this is mother cell. You could see this is mother bacterial cell, and a bud is developing. This is the bud that is developing, that is growing bud. And before that, what happened? DNA is double. This is DNA, this is another DNA is double. The process is called as replication, and now the bud detaches from its parent bacteria. Now both are forms individual organisms. This is how budding happens. This is the second one. What is the first one we have seen? Binary fission we have seen. The second one is going to be budding. And let us move on to the another one. The third one is called fragmentation. This mode of asexual division, and earlier both are also asexual divisions. This mode of asexual division is more common in where? More common in filamentous bacteria. Filamentous in the bacteria means that are long thread-like bacteria. In this mode, filament of growing cells get fragmented. That is, for example, you just see this is a thread, bacterial thread, and what happened? Fragmentation takes place here and here, here and here. Then what happened? This cuts here, and each cut will develop a single bacteria, and fragmented into small bacillary or coccoid cells. These cellular fragments eventually develop into a new cell. This is what is called fragmentation. And let us move on to another one. So far we have seen what? Three modes of bacterial division we have seen. One is called binary fission. The second one is called budding. Third one is going to be fragmentation. So out of these three, the first one, binary fission, is the common one, which is by using that one, bacteria grows a lot. Let us see the another one. Now we are going to see the another one is bacterial growth curve. What is this bacterial growth curve? There are generally 4 phases of bacterial growth. Now let me tell you that this bacteria, we are not observing this bacteria as a single unit. We are observing the population of bacteria. And when you are observing the growth of the population of bacteria, you generally observe 4 phages in the bacterial growth curve. The first one is going to be lag phase. This is what is it? Lag phase. And the another one is going to be log phase, the another one is going to be stationary phase, and fourth one is going to death or decline phase. So, and this curve here you are observing here, this is going to be, what is this one? This is going to be growth curve. We will explain this one later. And how many phases are there so far we have seen in bacterial growth curve? In bacterial growth curve there are 4 phases are there. One is lag phase, log phase, stationary phase, and death phase. And before we are going to see one by one, I would like to explain some important thing that is going to be, bacteria are very small in size. That is the first thing. We all know that one. Bacteria is very small. That is what we call it as microscopic in nature. Only we could observe them under microscope. That is why we are studying individual bacterial cell is impossible. Almost it is impossible. We cannot able to study the growing of individual bacterial cell. That is what we are studying, bacterial population, not the individual bacterial cell. We are studying bacterial population. So while you are studying, what you will get? You will get a growth curve, bacterial growth curve. You will get a growth curve. That growth curve is determined while you are observing bacterial population. So in order to get this bacterial growth curve, what you need to do? First of all, you need to prepare a fresh, suitable culture media. So for example, I am taking here a small flask. This is the small flask. Inside this flask I am having, what is that one? Medium. And now what I am going to do, small number of inoculum from old culture. Somewhere I take a few bacteria, taking that bacteria and putting in this flask, taking somewhere the bacteria and putting it into my new fresh culture media. That is called inoculation. A small amount of old culture, old bacteria is taken, and I just keep them in fresh culture media. This is called inoculation. This process is called inoculation. The small amount of bacteria that I put inside the fresh culture media is called inoculum. This is very important. This inoculum generally prepared from old culture. This is old culture. When you inoculate the old bacteria into a fresh culture medium, then what happened? Growth of cell is monitored. The cell's growth, how the bacteria is growing, that is monitored at a regular interval of time. For every hour, every half an hour, used to see how the bacteria is growing. Now what happened? The growth curve is plotted. Then once you are observing again and again at the time interval of half an hour, 1 hour, then you can able to plot a curve. Whatever the curve plotting is called growth curve. You will get a growth curve. How will you get a growth curve? Let us see. The growth curve used to have 2 axes. One is x-axis, another one is y-axis. X-axis and y-axis. Under x-axis, what you are going to take? X-axis I am going to take time. Whatever when you are taking readings for 1 hour or half an hour, whatever it may be, you take time. And on y-axis you take logarithm of cell number. This is important. The cell number in log you will take in y-axis. Then if you plot that one, what you will get? You will get a bacterial growth curve. You will get a bacterial growth curve that will be seen in the next slide. How that bacterial curve is like, we will discuss that one in the next slide. So as I told you, while you are observing every hour, half an hour, you will able to see 4 phases. The first phase is called What is that one? The first phase is called, what is that one? First phase is called lag phase. What is the another one? What is the another one? Log phase. What is the another one? Stationary phase. The last one, death or decline phase. Let us see the first one. What is the first one here? The first one is going to be lag phase. What is lag phase? No increase in the number of bacterial cells. So you just taken an inoculum from the old culture and inoculated that one into the new fresh culture. After you take reading how much the bacteria has grown, you have taken a reading, but that reading doesn't show anything. Why? Because there is no increase in number of living bacteria. That is called as lag phase. We will discuss in a lot about this particular phase, and he is talking about log phase belongs to this particular portion of the curve. And then another one is what? Another one is log phase. You could see log phase. Log, this is log. And another one, this is going to be what? Stationary phase. And this is going to be declined or death phase. And you could see what you could able to observe in lag phase. Lag phase, no, the bacteria growth is almost parallel to the x-axis. No increase in this direction. What is there in this direction? Logarithm of living bacterial cell. No increase is there in bacterial number. Later you could see in the second phase, log phase. In log phase, you could see the curve tends to upwards. That is an exponential increase in the number of living bacterial cells. You could see in the second portion of this curve. And third portion is stationary phase, where a plateau of number of living bacterial cells, the rate of cell division and cell death are almost equal. So birth rate and death rate are equal. Then what happened to the population size? Population size is parallel to the x-axis. Then the last one is decline phase. Decline phase indicates more deaths are there than cell division. That is why the number of bacteria, size of the population decreases here. So that is why the fourth phase is called, what is that one? Death phase or decline phase. We will see more information regarding each and every phase, whatever I explained here. Now what we need to see on y-axis, what we have seen on y-axis, what we have seen on y-axis, we have taken on x-axis we have taken time, on y-axis what we have taken? Logarithm of living bacterial cells. That is what we have seen. And next we are going to see more detail about log. Initially when we inoculate, that is what I explained, we inoculate old culture in a fresh nutrient media. That is what we have seen. The culture do not carry out its multiplication. That means no cell division takes place immediately. So then what happened? Here in the beginning, the cell growth remains steady. No population growing is here. That is what he is telling. The culture of microorganisms try to adjust the new environment. That is very, very important. New environment. So far, the bacterial cells are in the old culture. They are accustomed with the physical and biochemical environment in the old culture. But what you have done, you introduce them into a new fresh medium. Fresh new medium has different biochemical and physical environments. So it is now facing new environment, that is with new fresh nutrients, temperature, pH of the media. So that is what it is trying to adjust. The new bacteria is trying to adjust, isn't it? Whereas on another hand, the cells synthesize amino acids, protein, RNA, enzymes, and intermediate metabolic products which are further necessary for growth and cell division. That is very, very important. Now bacteria is not dividing, then we should not say it is not doing anything. It is doing something, being in the new media. What it is doing, it started synthesizing amino acids. It started synthesizing proteins, RNA, and some of the metabolic intermediates that are necessary for growth and cell division. That means it is start acquiring all the machinery for division. Okay, the time of lag phase, the time taken by the bacteria for its lag phase may vary from species to species due to various environmental factors. At the end of the lag phase, the bacterial cells start to multiply and cells enter in the logarithmic phase of growth, that is log phase. Once bacteria completes its lag phase, then it simply enters into the another phase called log phase. Let us see the log phase in the next slide. In logarithmic phase, also known as log phase, this is the second phase in the bacterial growth curve. We have already seen in the second phase this thing in the bacterial growth curve. Lag phase also called as exponential growth phase. Why it is called exponential? For example, this is x-axis, y-axis and x-axis, and this is what is called a lag phase, and then this upward movement of the curve is called log phase. And what we have taken in the y-axis, logarithm of number of bacterial cells. That is what this growth is called as exponential growth phase. Exponential growth phase. Why it is exponential? For example, if initially there are 2 bacteria is there, and this 2 bacteria after 10 minutes or so it converts into 4 bacteria. And after 20 minutes or so it converts into 8 converts into 16 to 32. In that way it multiplies. That is what it is, exponential growth phase. In this growth phase of bacterial cell multiply and carry out cell division. The rapid cell division takes place only in log phase. The cell division in this growth phase is constant and maximum. This is very important. It is maximum. The growth is maximum in this phase. Here the number of cells increases exponentially with the time. As time grows, the number of cells also increases. And another one, in this growth phase, all bacterial cells are small in size and biologically active, as they are constantly dividing. So into most biochemical and biological studies, bacterial cells from this exponential growth phase are used. So if you want to study any investigation that is related to either biochemical or biological studies, generally which phase of bacteria is generally preferred? The phase that is preferred is log phase bacteria. This is very important. And another one, if we plot a graph, already we have seen how we plot a graph. If we plot a graph of logarithm of a cell number against time on x-axis, it results in a straight line with positive slope. This is important. This is what is called positive slope. Positive slope. The multiplication rate and generation time of bacterial cell vary from species to species under different environmental conditions. What is this multiplication rate? And what is this generation time? For generation time means, for example, if 2 bacteria is there and if these 2 bacteria double, if 2 bacteria becomes 4, the time taken to double itself, its population, is called as generation time. How fast it is multiplying, that is multiplication rate. These are all will be discussed in the next video. let us see now. The multiplication rate and generation rate of bacterial cell vary from species to species. That means all species won't be same generation time and multiplication rate under different environmental conditions. And we come to the last point of this particular log phase. At the end of the log phase, the growth rate decreases. So growth rate is decreases, that is generation time of the cell increases. You could understand. So when the generation time is increases, it takes more time in order to double. After the end of the log phase, the bacterial cell enters in a stationary phase. These are the points, very important points we discussed so far. And let us move on to the another one. That is what is that one? Stationary phase. In stationary phase, the cells enter in stationary phase. The number of cells remains constant. I already been explained. Stationary phase, where birth rate is equal to death rate. So where you could see that kind of thing in the stationary phase, remains constant. When the birth rate equals to death rate, the size of the population remains constant. Generation. So because generation time of the cell is very low. And another one, it occurs due to various factors like why stationary phase is happening is explaining, like because of its depletion of nutrients, accumulation of toxic waste products. Generally, while bacteria growing, so it also produces some unwanted toxic waste products. Due to their toxicity, the bacteria also affected. That is why the growth rate is also decreases, and sometimes it may also changes its pH. And another one, due to all these factors in this phase, the growth rate is equal to the death rate, and the total number of cells till the end of the stationary phase remains constant. This is called as stationary phase. If you plot a graph of log phase of cell number against time, a straight line parallel to x-axis is formed. With the end of this phase, the death phase starts. You can see this is what is called log phase, and this is what is this one, lag phase, and this one is a log phase, and you will get a straight line. This is called stationary phase. This is the third one we have already seen. And let us move on to the another one, that is going to be last one, death phase. Where in death phase, we will see in death phase of the growth, the bacterial cell start to die. Here the death rate is higher than growth rate, and the death rate increases to maximum at the end of the death phase. There are various factors responsible for this phase, like exhaustion of nutrients and accumulation of toxic waste products. That is what we discussed, and drastic changes in the pH of the media. If you plot a graph versus what is that one, a log number of cells against time on x-axis, a straight line with a negative slope. This is what is that one that is having positive slope? That is log phase. What is that one having negative slope? That is death phase. Will be observed here with the death phase of bacterial growth curve. We have already seen that one, bacterial growth curve. Phase is called lag phase. Lag phase, the first, when you take a old culture and incorporate that, when you inoculate that old culture into the new fresh medium, then the cells tries to adjust the new environment. It won't divide immediately, and whatever the graph, when you observe this bacterial population, the graph, when you observe the graph, the bacterial curve almost parallel to the x-axis. And later, and now what happened, bacteria already been adjusted to the new media, and now it acquired all its machinery like amino acids, proteins, new metabolites, all the machinery, all the weapons it gathered, and now it starts what? it starts cell division. That is what we observed in the log phase. This phase is also known as exponential phase, where you could see rapid growth of bacteria, rapid cell division you could able to see in lag phase. And the another phase is, what is that one? The another phase is stationary phase, where once bacteria dividing further for 1 day or 2 days, what happened inside the media, nutrients becomes depleted, and bacterial toxic waste products are accumulated, and the pH also changed inside the media. There are these reasons makes the bacteria won't divide. If the bacteria won't divide, or the generation time becomes much higher, then what happens? The bacteria stop dividing and the population size it won't go further high. So birth rate is equal to death rate. That is what we could see in stationary phase. A plateau in number of living bacterial cells, the rate of cell division and cell death becomes equal here. So the last one is death rate. As we have seen, there are no nutrients inside the medium, pH is very different, and now bacteria won't divide further, and what happened? It dies simply. When the bacterial size is lower, what happened? The population size is also decreased. That is what we see as a fourth phase, that is called death or decline phase. With that, I am concluding this video, and hope you all doing well. We'll see in another video. Bye.